Patrick, and thank you very much to the organisers of today for the opportunity to speak. It's really brought me back to my Ulster University roots as a nutrition graduate, where I first gained an interest in vitamin D working with Emer McSorley and the teams there studying autoimmune disease. I've brought that interest forward to my research now on cancer epidemiology. And today I'm going to be providing insights from a study design that we call molecular pathology epidemiology. So measuring vitamin D exposure in a slightly different way to try and improve the plausibility of the associations that are often reported in epidemiological studies. And I just want to acknowledge that a lot of my work is possible thanks to a fantastic infrastructure that we have in Northern Ireland. So in Northern Ireland, the population of 1.9 million people are all serviced by one of 5 health and social care trusts through the NHS point of free healthcare system. Those 5 trusts are serviced by 4 pathology labs. And thanks to the infrastructure of the Northern Ireland Biobank, we can apply to access archived tissue of cancer patients who've undergone surgery, for example, to do studies and analysis in a way that is relatively light touch in terms of ethics. You apply through the Northern Ireland Biobank, and this is all done in an anonymous way that you don't actually have to contact the patients themselves. One of my roles is as Deputy Director of the Northern Ireland Cancer Registry, which is what we use to identify the patients in particular. And then the third component of this is my colleagues at the Precision Medicine Centre, which enable us to do some of the tissue microarray work that I will describe. So what is molecular pathology epidemiology? And this is a figure from Shuji Ojino's group in Harvard. Shuji coined this term approximately 15 years ago. But essentially, a lot of the work in my area is traditional epidemiology, where there's lots of studies showing a variety of lifestyle factors that are associated with cancer risk or cancer survival, including vitamin D as measured through blood serum status. You then also have a lot of traditional molecular pathology studies looking at a variety of cancer biomarkers for prognosis. But there's very, very few studies that try to bring these two worlds together. And really, that's a shame on our part as researchers. We need to be doing much more to bring together lifestyle factors and patient exposures with what we know about tumour biology in order to investigate associations with prognosis and treatment. And I'm sure lots of people here will be aware that vitamin D has many benefits, and that includes colorectal cancer risk, for example. There's been lots of studies showing associations between serum vitamin D status and a reduced risk of cancer, including colorectal cancer. And this is just one meta-analysis from over 10 years ago now, but there's been many studies published since. But there's lots of unanswered questions. Is there an association between vitamin D and colon cancer progression rather than risk? The biggest one to my mind is it really vitamin D? We get a lot of criticism that is it actually vitamin D, or is this just a proxy for other confounders and a measure of healthy lifestyle? And to be honest, serum vitamin D measures are never going to help us to answer that question. We need to look a bit further. I'm also interested in are there subgroups of individuals, and in particular, in my case, cancer patients who would benefit most from vitamin D supplementation studies or other ways to enrich. And so I really believe that molecular epidemiology studies are going to help us answer these questions. So a lovely study published 7 years ago now from Lena Zgaga and colleagues in Trinity did provide further evidence that vitamin D in the serum should be beneficial in terms of cancer survival. So patients with the highest levels of vitamin D are shown by the purple line and had improved survival compared to those with lower levels. And this seemed to be stage specific, so it was particularly prominent in stage 2 patients. with also some evidence in metastatic patients. Sorry, not in metastatic patients combined. If we look at tumor vitamin D receptor status then, so actually seeing within the tumor itself, what's the expression of vitamin D there, and is that associated with survival? The initial results were quite disappointing. So again, this is a study from over 10 years ago from Shujio Jaino's group. And it did not show that there was any significant association between tumor vitamin D receptor status and survival in colorectal cancer patients. But what I would draw your attention to is their choice of antibody, the Santa Cruz antibody that they use to try and measure vitamin D receptor expression. We tried to replicate that in our labs and couldn't. And in fact, we went through 5 choices of antibody before we could get one that we were satisfied was actually measuring vitamin D receptor expression. So the jury was still out at that stage. So for our work, what we did was we used the cancer registry to identify all colon cancer patients who were diagnosed at stage 2 and 3 because they would have undergone a surgical resection. And this was all patients within 2 of the health and social care trusts in Northern Ireland over a 5-year period. And we had follow-up information for up to 10 years for these patients. We had 661 patients included in the study. And so for the biobank, that meant they had to go and retrieve all the slides relating to those resection specimens. And those slides then needed to be reviewed. And that actually led to a review of over 16,000 slides for our entire study. So this is not a light undertaking by any means. The slides then are put over the tumour blocks themselves, and through the Precision Medicine Centre, tissue microarrays created. And what this does is it enables us to do really large scale studies of vitamin D receptor expression. And it just so happened at that time, QPath was also being developed at Queen's, allowing us to do large scale digital analysis. And we were involved in some of the validation of that. So we found that the VDR antibody from cell signaling was actually the most reliable measure. And this just shows you one example of a tissue microarray. This study resulted in over 2,000 cores that would need to be scored in order to look at both the intensity and expression of vitamin D receptor. We did also extract DNA and look at a variety of vitamin D related SNPs in our study, alongside a measure of MSI status and other KRAS and BRAF mutations, which are known to be a prognostic in colorectal cancer. And what I will say is this is a long undertaking doing these types of studies. I should probably update this. 10 years later is probably more accurate now, but it takes time to build up this resource. And at the end of that, what we found was the first biological evidence that tumour vitamin D receptor expression was associated with a reduced risk of death in colon cancer patients. So that was really encouraging, but at the same time, we did not see a dose response association here. The strongest association was in the middle, which is always a bit dubious as an epidemiologist. We'd love to see a nice dose response relationship. So overall, we were encouraged by these results. We didn't see any associations when we looked at the corresponding vitamin D receptor expression in adjacent normal tissues. So this was only in the tumour itself. We did see that one subgroup of patients with microsatellite stable tumours where the associations seemed to be stronger. And we didn't see any significant associations by the SNPs, but I think our study, even though we had 661 patients, it was probably a bit underpowered for the genetic analysis. We have then applied this in esophageal adenocarcinoma, which is a cancer of considerable concern in Ireland. We have some of the highest rates in the world. And in this, we did see a strong inverse association. So patients with the highest vitamin D receptor levels had the lowest risk of death from esophageal cancer. This study only included 130 patients. And one of my major focuses at the minute is trying to replicate that and expand that hypothesis. So we're undergoing a TMA creation within the UK Occam's consortium, which includes over 4,000 patients. We're hoping to create a TMA for 1,200 of these. And that's the focus of a CR UK fellowship that I'm currently working on. So hopefully I'll be able to present to this group again in a few years' time with some updated results from that. So in conclusion, tumour vitamin D receptor expression was significantly inversely associated with colon cancer survival. So this is the first biological mechanistic evidence that it seems to be vitamin D that's beneficial and not just confounders or a marker of healthy lifestyle. The choice of VDR antibody is really important. And also I think the choice of cutoffs from a statistical point of view is important given the lack of dose response that we saw. We've seen similar results in esophageal cancer and we're undergoing further research from that. But our approach to these study designs is really strong because it's population-based and it's representative. It's generalizable to all cancer patients, not just those who volunteer or are selected for participation in trials. So I just want to thank a huge array of individuals, and particularly the late Professor Liam Murray and Cancer Research UK, who have funded this work in colon cancer, and the UK-wide team that are involved in the esophageal cancer project currently ongoing. So thank you for listening, and I look forward to the Q&A later on.